Hi, everyone. Dr. Wiseman here again with you for part two. This is specifically part 2A of the for week one material. This is scheduled for session two. So we're going to be seeing this on the second day of the week. So we finished the last lecture on the anatomy and physiology, the organs, if you will. Now we're going to go down into the sort of the subcellular level. Why do we digest things? Well, the first reason we digest things is that it's very difficult to get things across the plasma membrane unless they're very, very small. Large objects, even microscopically large objects, if you will, things you couldn't see even hardly with a microscope, are still too big to cross the plasma membrane. So that's one reason. We want to get these things down to a size that they can be relatively easily transported across the membrane. And secondly, we are going to make those molecules into new things. They serve as the building blocks of the things that we actually want inside and around our bodies. In order to achieve that, we use these things called enzymes, and they're mostly protein. Sometimes they have a little this or that helps it, like vitamins and other things that help the protein do what it does. And these things act as catalysts. That is, they make chemical reactions possible in physiologic conditions. For instance, you can oxidize glucose. Glucose, that's what we do in order to make ATP. But if you do it using a cigarette lighter, that glucose has to be raised to somewhere around 750 degrees Fahrenheit. Well, that's not a physiological condition. Membranes and material would not last at that level of temperature. So we use enzymes in order to allow these things to occur at physiologic conditions. And what enzymes do is they act on substrates. So here we have this purple molecule here, and here's an enzyme down here, and you can see there's kind of a binding site. They call that the active site. That substrate will interface with the enzyme at the active site, and in this case, this enzyme is breaking that molecule down into two smaller products. This is what we would call a catabolic enzyme. It's breaking something down. So, as we mentioned, enzymes are mostly protein, but they do sometimes have additional molecules, additional things that allow the enzyme to do what it does. Sometimes it's a metal ion, sometimes it's an organic molecule. We call these things coenzymes and cofactors. We'll talk a little more about that in the upcoming slides. Some enzymes that you're going to want to keep in mind. Again, the key here is to simplify, come up with a haiku, that's what I like to say, a 3 to 5 word phrase that you can recall what each of these enzymes do. For instance, we have amylase, and that's found in saliva, but we also have a pancreatic form of amylase. This one breaks down long chain carbohydrates into smaller ones. In particular, it'll break it down, if left long enough, into what are called simple sugars, monosaccharides. Glucose, maltose is another one. So, for instance, if you go to a Mexican restaurant and you're really, really hungry and you have that first, tortilla chip that you get on your table, tastes really, really good. It's because the amylase in your saliva is breaking down the starches into shorter sugars. And those shorter sugars are what give the interesting and unique flavors to foods. There's one called pepsin. This one's in the stomach. We've talked about pepsin before. It is a protease. It breaks down proteins into smaller amino acid chains. We call them polypeptides in some cases, dipeptides, that would be 2 amino acids, tripeptides, that would be 3. And so pepsin does that, and it works well in that low stomach pH. Then we have 2 more proteases, but these are coming from the pancreas, so these work at a higher pH than pepsin does. They're called trypsin and chymotrypsin. Now, this word zymogen, now it's a fancy word, right? What that means is that when they're made, they are inactive. A zymogen is a precursor. Remember when I said ogen means precursor, and so it's a precursor of an enzyme. A zyme It is the ogen as a precursor. So we have these trypsinogen and chymotrypsinogens. Those are the zymogens. And when they're activated, once they get into the digestive tract, they will break down proteins. Then we have lipases. All right, these are made by the stomach and also by the pancreas. You can see the pancreas has a lot going on. What we're going to do is we're going to be breaking down fats. Fats are primarily a molecule called a triglyceride. It has a glycerol, looks kind of like a letter E, and then it has these heavy carbon and hydrogen. They're called fatty acids. They look like these little zigzag molecules. I'll show you one here in a second. That's where all the energy is in the fat, is in those bonds in the carbon and hydrogens in the fatty acids. Then we have nucleases. These break down DNA and RNA into their individual nucleotides, again made by the pancreas. And this is also serving an important anti-pathogen role. These enzymes will degrade DNA and RNA it encounters. That includes from bacteria or other potential pathogens. And then we have an assortment of what we call brush border enzymes. Remember that we have those villi, and if you see lots of villi, it looks kind of like a brush. Well, these enzymes are embedded in the plasma membrane of these villi, and they'll do all of the above. And they'll break down carbohydrates and proteins. Again, there are just so many different versions of them that we don't want to have time to do them. So nucleases, lipases, trypsin, chymotrypsin, pepsin, and amylase. A little background about sugars, and you should be able to sort of recognize them for what they are. I'm not going to tell you is it a glucose, a lactose, or fructose, but you should be able to recognize a sugar. And here's how you do it. Here's the secret. See this O here? That's an oxygen. Go clockwise. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6. All right, so if O is 0, 1, 2, 3, 4, 5, 6. Here's the O here. This one's a little bit weird. 1, 2, 3, 4, 5, 6. These are all what we call 6-carbon sugars or hexoses. Glucose is the predominant one that we use for energy. These other ones we often convert into glucose before we metabolize the glucose to make ATP or that sort of thing. So these are hexoses. That means they have 6 carbons in that sugar. It's a ring, and look for that O in the ring. That's the key. All right. You can see this one's kind of a 5 here, but it's got that 6 one out here. That's still a ring, though, with that O there. We also have what are called pentoses, and the reason I bring these up, you probably don't see these in your original PowerPoint, but I think it's a good idea, just again, to reinforce how you would recognize it as a sugar. So here's the O, 1 carbon, 2 carbon, 3 carbon, 4 carbon, and then the 5th one. Same thing here. Here's the oxygen, 1, 2, 3, 4, 5. These are 5 pentoses. These are pentose sugars. And you have ribose and deoxyribose. These are really important for DNA and RNA. R for ribose and deoxyribose for deoxyribonucleic acid. When we're doing breakdown of sugars, oftentimes you'll see the ose, that means sugar, in there. Oftentimes what we're talking about when we're talking about our food is what we call a disaccharide, 2 sugars. So 2 hexose sugars linked together. For instance, if we have sucrose, which is your normal table sugar that you use to sweeten your coffee or tea, you have an enzyme called sucrase. It's specific for sucrose, and it breaks it down into a glucose and another simple sugar called fructose. Lactose, we find this often in cheeses and milks and so forth. What you have here, you have an enzyme called lactase. If someone is lactose intolerant, it generally means that their body is not producing enough lactase enzyme. And so the lactose goes into the digestive tract, doesn't get broken down, and then the bacteria are more than happy to break it down for you, leading to large amounts of gas, which leads to distension and pain. But if you have lactase or you take lactase, if you're lactose intolerant, it'll break that lactose down into glucose and another simple sugar called galactose. Maltose, I happen to love malts like that you get at the ice cream shop. Again, it has kind of a creamy flavor to it. You find it in milk products and so forth. This is a disaccharide of glucose and broken down by an enzyme called maltase. So here's our maltose, 2 glucoses. And if we break that down into a single glucose, we'd use that maltase to do it. Like I said earlier, fructose, galactose are eventually generally converted into glucose before being metabolized for other purposes. Now a little bit about proteins. The subunit, the monomer of a protein is known as an amino acid. It has this kind of structure here, a nitrogen, carbon, carbon. This left-hand side, that's the amine. That's what we call the amino chemical group. And then this COOH is a carboxylic acid, or carboxyl as it's shown here. That's why we call these things amino acids. We have about 20 or so amino acids in our body that we use to make protein. There are some other amino acids out there that are not used for protein. But in any case, what makes each one of these different is this group right here, this R. This R is a variable group. And it's, like I said, there are about 20 different things that can link in there, give or take, depending on if you're talking about proteins or other things, that link right here that make each individual amino acid different, like these different colors here. All right. And we link amino acids together and we create this three-dimensional structure, sometimes multiple subunits. All right. The bond that holds amino acids together up here at the top is called a peptide bond. It's a covalent bond. And so we link an amine group to an acid group and so forth. And that's why amino acids are the subunit. In previous lectures, we'd talked about macronutrients and micronutrients. Well, the macronutrients are the things that we have to break down. Micronutrients are generally imported without too much modification. Occasionally you need some help here and there, but most of the time they're transported as is, whereas macronutrients are often way too big, way too bulky in order to get across the plasma membrane in their original form. So for instance, over here on the left, we have carbohydrates. They have to be broken down into generally simple sugars, monosaccharides, and these are some hexoses here, glucoses, all right, in order to be transported in. And we use those carbohydrates. We certainly, everybody knows carbs are for energy. The way we make energy from those carbs is generally through the synthesis of a molecule called ATP. Proteins can also be used for energy, especially in times of caloric deficit. So if you lack enough carbohydrates to support ATP synthesis, you can use proteins. And proteins are broken down into their individual amino acids, then they're transported in. And then those amino acids are then, if they're not used for energy production, about 10% of them are, the other ones would go into the creation of new proteins. Then lastly, we have the fats. So fats have to be broken down from these large globules. Sometimes they get emulsified using the salts from bile. We also will break down the fats with lipases and to make them into smaller molecules, free fatty acids. Another structure that's a glycerol and one fatty acid called a monoglyceride. That way we can bring them in and then reassemble them to create fats or do other things with those. So energy is certainly part of that. Yeah, everybody knows that. We can store it in a structure known as adipose tissue, fat tissue. But these things are also used in things like hormones. We generate, using fats, we generate cholesterol, and then cholesterol can serve as a precursor for steroid hormones. Now, fat absorption is a little different than the other types of absorption because what we do is we get these droplets of fat. All right. And then these things will form what we call these chylomicron balls, if you will. And these have triglycerides, and sometimes they're inside, but generally they form this sort of like a almost like a plasma membrane like structure. All right. And these have to be absorbed. And so they are absorbed by the special lymphatics in the digestive tract. I've mentioned these before. They're called lacteals. Those are the lymphatics that go up into the villi. And then those flow into larger lymphatics, which are then returned into the bloodstream near the subclavian vein. And then through the bloodstream, then the cells will take up these fats through in the form of triglycerides. Anything left over gets processed by the liver into either other structures or is dispensed in the bile. There is an enzyme that we find in the blood called lipoprotein lipase. So as we take these things in via the lacteals and lymphatics, what happens is when it hits the blood, there will be further enzymes to break down. One of the things that happens, if you think about heart disease and athero and arteriosclerosis, what happens is if you have persistently high levels of triglycerides and or another lipid called cholesterol, these lipases get overwhelmed. They just can't break down these materials fast enough. And those molecules end up distributing in little eddies in the blood vessels. And there are lots of little eddies in the coronary arteries where those things can reside, and the heart doesn't have much of a sense of humor about blood flow. So if those get clogged there, myocardial infarction. As we said, macronutrients get broken down. Micronutrients are generally not broken down, if at all. Maybe sometimes they get modified a little bit, but most of the time they're usually in their original form that was brought in during intake, either through fluids or solid food. These micronutrients, one of the most common things that they are used for is as cofactors. That is, they help the enzyme form the proper shape to achieve an enzymatic function. Sometimes they're used in protein synthesis as well. What they do is they basically assist enzymes. For instance, vitamins are organic molecules, and many of these are cofactors. They call them coenzymes. We have fat-soluble vitamins, A, D, E, and K. A has been in the news quite a bit lately because of the measles outbreak. I want to talk about that in just a moment. The thing about fat-soluble vitamins is that you can generally take too many of these. You don't lose these very quickly. On the other hand, the water-soluble vitamins, that's the C vitamins and then the various B vitamins, these ones you kind of have to take on a daily basis because you do lose them to the urine. The fat solubles, not so much. As long as you have a relatively well-balanced diet, you're going to get plenty of these and you don't need to take extra. And here's the risk. There are a lot of people right now that are going out of their way to get vitamin A and or cod liver oil, which happens to also be high in vitamin A. And vitamin A is really, really toxic. Vitamin E is also toxic too. Your body does not deal well with elevated levels of A and E. All right. If you take too large a quantity, and if people are taking vitamin A and cod liver oil, there's going to be incidences of vitamin A toxicity showing up in those populations where people are trying to use these remedies rather than vaccination. What happens is they build up in the liver, they hamper its function, just like ethanol can cause cirrhosis and fatty liver disease and even death. Minerals, on the other hand, are inorganic. That means they don't have carbon in them. And a lot of these are metal ions, calcium, zinc, magnesium, iodine. They serve as cofactors for enzymes, but they also are important for the osmolarity of the blood and the fluids, sometimes for other purposes. Iodine is particularly important in molecules called thyroid hormones. In any case, take a look at tables 24.2 and 24.3 for further details. Take a look at those. These things are super important for nutrition, but we have to have the right kind of nutrition. More is not better. More is not better. So when we digest food and it's being passed through the small intestine and then into the large intestine and eventually out, what happens is after absorption, that blood generally flows through what's called a hepatic portal vein. Now, what does that mean? A portal system, let me back up for just a second. You have your artery here, becomes an arterial blood vessel and so forth. You go through an organ, then you become back into a vein, and it generally goes back to the heart at that point. A portal system is different. It has a capillary bed, and then it condenses again into what we call, by convention, a vein that then breaks into a second capillary bed before it returns back to the heart. And there are actually 3 dedicated portal systems that we know of in the human body. In this case, this is the hepatic portal system that is going from the digestive tract, the stomach, spleen, pancreas, small and large intestine, all right, through these mesenteric arteries and veins. That's the portal system that goes to the liver, where the liver will begin screening out certain compounds. It will, you can say it is a sort of detoxification. That's true. That's sort of the intent. But what it's doing is it's screening through some of these things. And so what we see is we see about 80% going through that portal system, if you will, in order to screen things out that the body does not want or need to be secreted in the bile. And then back into the regular circulatory system. So that's the hepatic portal vein. As much as I hate to do it to you guys, we do need to talk a little bit about biochemistry. And oh my God, how can I do this? How can I do this? Well, first of all, you don't need to know every single thing in this chart. It looks super duper complicated, and it is, but nobody has this entirely memorized from scratch, unless you're one of those photographic memory type people. The thing that we're going to try to do here is show you some basic aspects of this that we think are essential for primary care. All right. And so when I say Achtung, which means warning, go slow. Go slow. Take it slow. You will get it over time, but you can't get it by being passive. If you just stare at it and you're not actively thinking about it, you're not drawing it, you're not talking about it with other people, you might as well be slamming your head into a brick wall. There are things in this life that cannot be learned passively very easily, if at all. And this is kind of one of those things. So I'm not trying to scare you here. You can do it. We know you can, and students do it all the time. Thousands and thousands of students do this every year, and you can too, but you've got to go slow and you have to be active. First of all, some concepts that we have already talked about, but we want to make sure that you keep these sort of in the front of your brain. We break things down using catabolic reactions, and these things often will release energy. We're going to be using a reaction. We're going to show you a diagram of it later on called hydrolysis. Lysis means breaking down. So if you see lysis, that means that whatever is happening, something is becoming smaller or energy is being released. And I'm doing this one first because catabolism releases energy and provides material for building up, which we call anabolic. You might have heard of anabolic steroids. That causes increased muscle mass. Anabolic reactions require energy in order to make larger molecules out of smaller ones. We're going to talk about these terms here, oxidation and reduction, or otherwise known as redox, and we'll explain that in a second. There's a simple concept for you to have some basic understanding. Almost all of these reactions, to one degree or another, can take place without enzymes, but they require energy that then could be destructive to cells. So we use enzymes in order to facilitate the reactions in physiologic conditions, at temperatures, at pH, et cetera, that will not destroy cells and tissues. And so we use enzymes to do these anabolic and catabolic redox reactions. Most of these reactions, like that chart you saw on the previous slide, happen inside the cell. Many happen inside a mitochondria. That's where we synthesize ATP from glucose. Things happen generally inside the cell, but not always. There are things that happen outside the cell. For instance, we had a catabolic reaction we just talked about a few minutes ago, that lipase that flows through the blood that breaks down lipids. It happens extracellularly. And these reactions sometimes can work without oxygen and sometimes require oxygen. In the mitochondria, what we're doing is we're generally making ATP using oxygen. That's why we call it aerobic. Most reactions don't require oxygen. So most reactions in the cells are, in fact, anaerobic. They don't use oxygen. So here's our representative catabolic reaction. It's very, very common. It is one of the, if not the most common reaction that we see in the body. And it's called hydrolysis, where you use water. This water molecule gets split into an H and an OH. The H going on to one of the products and the OH going on to the other. For instance, glycogen is a polymer of glucose. So if we have multiple glucose subunits in a chain, we use hydrolysis in order to break off the glucose from that glycogen, and in the process, that H2O is destroyed. Again, another very, very simple concept that many of you will no doubt be somewhat dismissive of, but since you're in this class, this is relevant for your grade, which makes it relevant, so don't dismiss it, at least for the time being. That is what we call redox. That is, I say, oxidation in purple and reduction in green. What happens is when you add an electron to a molecule, the charge gets lower. That's where the word reduction comes from. It is a reduction in charge. Maybe this B here had a charge of 0, and when you added the electron, it became a charge of -1. So it has been reduced in charge. That's where that comes from. But in any case, Leo is a German. If you lose an electron, like here, this A becomes an A positive. It's lost an electron, so now it's got a positive charge. It has been oxidized. That's because oxygen frequently steals electrons from other molecules, right? For instance, glucose, all right? Here, this B started out as a charge of 0, but because it gained an electron, that's the German. So when you gain an electron, you are reduced. So A is oxidized and B is reduced. Just start there. And again, I know that this will feel really not in your lane, but there are reasons why we teach this, and so have some faith for me. Leading off on that previous slide, what we are going to do with glucose up here at the top, in this what we call cellular respiration, is we are going to oxidize glucose into carbon dioxide, and eventually we are going to generate water. The oxygen here at 6 is being reduced, and we literally create water. This is metabolic water. So the glucose is being oxidized and the oxygen is being reduced. And this occurs in 4 main steps. The first step up here happens actually out in the cytosol. It's outside of a mitochondria, and it is called glycolysis. In periods of extreme 100% effort, where you're doing a pull-up or something, where you're using your maximum force, you begin to feel the burn a little bit. What's happening there is that you're going into what's called a glycolytic energy production. You break down these the glucose into a molecule called pyruvic acid, but you generate some ATP, and this is really, really fast. So in times of desperate need, like in maximal exertion, what happens is when you go, quote unquote, go anaerobic, you're only doing this. But in periods where you have high levels of oxygen, that pyruvic acid, also known as pyruvate, gets moved into the mitochondria, and that's step 2, where we create something called acetyl-CoA. So acetyl-CoA then feeds into the rest of the ATP production. We have the citric acid cycle, or also known as the Krebs cycle. This is where most of your carbon dioxide comes from. You're not making carbon dioxide over here. You're making carbon dioxide right here at step 3. So we got the Krebs cycle, citric acid cycle is also known as. And then lastly, we have the electron transport chain. And this is where the oxygen comes in. So we make carbon dioxide over here, and we process oxygen right here. And this is where we make most of our ATP. You can make a little ATP here and here, but it's not enough to do anything other than serve as a sort of stopgap measure under maximal exercise. So those are the 4 steps: glycolysis, making acetyl-CoA, Krebs cycle, and electron transport. Now, we can use glucose to do this. This is a carbohydrate, but we can also use proteins or fatty acids when we don't have a lot of carbohydrates available. And so we can actually, there are separate entries into this pathway using proteins and fatty acids. That last step, electron transport oxidative phosphorylation, that's where we make most of our ATP. That's what the one that requires oxygen. And you can see we make 28 or so. It varies a little bit. All right. That's the final step here. Glycolysis and Krebs cycle both generate a little bit of ATP. You can see you can make up to 4 here. All right. That is known as substrate level phosphorylation rather than oxidative because they don't require oxygen. So substrate level here, substrate level here. This is where you're using an enzyme to transfer a phosphate onto the ATP that doesn't require oxygen. So this is a little bit of a busy slide. The key thing is you can't just look at it. You have to walk your way through, and we're going to start in the middle. Carbohydrates, primarily glucose. We break those carbohydrates down into simple sugars, and glucose is a big one, or we convert other sugars into glucose. Glycolysis to make pyruvate. Then we make acetyl-CoA, and that goes in through the Krebs cycle. Then we hand off electrons in electron transport to make water from oxygen, and that's where we make a lot of our ATP. All right. But again, proteins can come in from different directions here. The amino acids can be converted into a carbohydrate, or they can insert as acetyl-CoA directly into the Krebs cycle. The same thing is true with lipids, where we can break down glycerol that way, or fatty acids through a process called beta-oxidation, which we will talk about here a little bit later. You're going to hear a term in the clinic called ketoacidosis. This is where what happens is your body is unable to absorb sugar into the muscle or other tissues, and so they become heavily reliant on these ketogenic amino acids for energy. And so what you get is you get a lot of these ketone bodies that then cause ketoacidosis. All right. Your liver and your kidneys can both generate glucose. It's a process called gluconeogenesis, where they can convert amino acids into a sugar-like molecule that then goes through glycolysis and then and down the pathway. So work this pathway first through the previous slides, and then you can kind of see how the other things fit in a little bit. And we're going to talk about beta oxidation here in a little bit. We'll talk a little bit about amino acids here in a little bit as well. I know it's scary stuff. I know it's something that's new to you and that many of you will question the relevance. It's important for nutrition. It's certainly important in certain types of conditions like diabetes, obesity, and so forth. So it will be useful at some point. It just isn't something that you normally see in your mind's eye when you're early in your career. Now I'm going to tread very, very carefully through here. Going to tread very, very carefully through here because this is heavy stuff, and so we're just going to just kind of do a little bit of an overview here. What you can do is that you can change amino acids into either acetyl CoA or glucose-like molecules called transamination, or there's another process called deamination where you get rid of the amine, and those then go into either the citric acid, or they call it the Krebs cycle, or you can be going through a separate similar cycle called the urea cycle. If somebody has gout, if you ever hear of gout, gout is an issue where you have such a protein-rich diet that your body is transaminating and deaminating, and those things go into here and they form uric acid, and you form so much of it that your body can't get rid of it quickly enough, and those deposit as crystals in the joints, leading to gout. So a little bit about nutrition here as well. We're not going to get into the nitty-gritty here. What we're doing is we're changing an amino acid here, or we're getting rid of the amine and creating a glucose-like molecule. A bit of a summary here. When we have material coming into the body postprandial, that is to say, while we're absorbing things, we'll go through glycolysis. Sometimes we'll generate glucose. Sometimes we'll make fatty acids and we'll generate fat. We'll certainly synthesize cholesterol and other types of proteins, and also process, 5 carbon sugars. Post-absorptive, this is generally, post 6 hours post-absorptive. What we do is we will break down glycogen into glucose. We will create glucose, and this is generally in your liver and kidneys, called gluconeogenesis. We will break down fats, and we will oxidize those fats to make acetyl-CoA and make ATP. And we'll also generate ketone bodies as we're at transamination and deamination processes to make glucose-like and acetyl-CoA-like molecules. Lots of people know that fats have lots of calories. We know that from our daily, you hear about that in the news and, public life and that sort of thing. Why are lipids the most energy dense? That's because they have lots and lots of covalent bonds that can be used to harvest energy. So here is what we call a triglyceride. This is the key component of fat. You've got this sort of letter E structure here. This is the glycerol. This is a triglyceride. And these molecules here, this double bond O, and then these zigzag, those are carbon, there's a hydrocarbon chain here. That's a fatty acid. 3 fatty acids, a triglyceride. Now, these two down here have double bonds in them, which means that they are unsaturated fatty acids, as opposed to this one at the top, which is a saturated fatty acid. Now, one more thing here. You'll see that there's the Greek letter alpha right here and a Greek letter omega. You ever hear of an omega-3 fatty acid? Okay, 1, 2, 3. There's an omega-3. This one's 1, 2, 3, 4, 5. This is omega-6 fatty acid. So if you hear that in terms of nutrition, that's what they're talking about. It's where the double bond is near the tail. The omega end is the downstream end. And so if it's an omega-3, it's 3 carbons upstream from the end, that sort of thing. But let's get on here. So if we're moving, triglycerides, generally what happens is they get processed in the liver into these molecules called very low density lipoproteins. And so what happens is these triglycerides get glommed onto these sort of carrier proteins. That's why we call it a lipoprotein. It's got a triglyceride and protein. Very low density lipoprotein and a low density lipoprotein. Now, we're not going to get into the technicalities that cholesterol is another form of lipid as well that we will sometimes deal with. The remnants after metabolism is returned to the liver via what's called a high density lipoprotein. All right, high density. And so triglycerides that's what this molecule is right here. These numbers, by the way, represent the normal value that we should see in the blood. 100 to 130 milligrams per deciliter of these guys, 35 to 60 of these guys, and then about less than 150 here. If you've got elevated triglycerides, you'd be above 150. Now on to how we generate ATP using this process called beta-oxidation. You remember in that previous slide, we talked about the alpha carbon and the beta carbon. And then out here would be the omega carbon way out here. All right. So what happens is we metabolize the triglyceride into this sort of CoA molecule, and it's called palmitoyl. And I don't want to get too crazy in this. That has to do with the number of carbons here. All right. But it has that a CoA, which is the same as acetyl-CoA. That is coenzyme A. And what happens is you break off this bond between the alpha and beta using an enzyme, and now you have these two carbons here, and that becomes acetyl-CoA that can then feed into the citric acid cycle, also known as the Krebs cycle. So you can have acetyl-CoA going directly in. You don't have to do glycolysis. You can do beta oxidation where you break this bond right here. This becomes acetyl, all right? When it's this long fatty acid, it's palmitoyl, palmitic acid, all right? In any case, that now is two carbons. That's acetyl-CoA, and that goes into the mitochondria. And so this beta oxidation process happens in liver, it happens in kidney, it happens in heart. You can't really do it in the brain, but it can do something sort of like it. Not going to get crazy with that. Again, you take this off of the glycerol, you attach the CoA, and then you hydrolyze that bond between the alpha and beta carbon to make acetyl-CoA. And if you have excess acetyl-CoA, you have more than you need, what can happen is you generate ketone bodies. So here's beta oxidation. We just talked about it. Got acetyl-CoA in here. We're going to get rid of that CoA. We're going to link it into some stuff and turn it into things like cholesterol or some other things out here. I don't want to get crazy with this. This right here is the first molecule in cholesterol biosynthesis, just to make a long story short. And that's it for today. I realize those last several slides are going to push you pretty hard. Take them slow. Work at it a little bit. Don't be passive. If you stay passive, you will not stick. I promise you. If you believe in your heart that it is not relevant to your life, well, take your chances, because you won't pass this class if you don't take the material seriously, even when you're not entirely sure that it's relevant. I know I'm getting a little strident here. I don't want to presume that you don't want to learn this, but I do realize that and I've seen it in well over 1,000, teaching thousands of students now, that attitude is everything when it comes to this material. So hang in there, take your time. It will be worth it, I promise you, down the line. There's no knowledge that you learn that you wish you hadn't learned, unless you're like a spy somewhere and then somebody wants to take you out, if you know what I mean. No knowledge is bad knowledge, especially when it comes to nutrition and well-being, even if you don't become a care provider. So good luck, and I will see you in part 2B soon.